Our second Nobel Prize laureate, Paul Modrich, he has spent his life trying to figure out how these mismatches, how they are corrected. And he has found a system which we now know as the mismatch repair system. And this repair system contains a number of different proteins. And what these proteins can do is that they can recognize a mismatch. You see this red-blue thing here is supposed to be a mismatch. It can recognize a mismatch. And then it can also recognize which of the two strands that are newly synthesized. Because you just want to correct the strand that's newly synthesized. You don't want to change the old strand. And then you cleave the newly synthesized strand and you take away the piece of DNA containing the mismatch, and then the DNA is synthesized again and we get the correct strand. This is a very important mechanism that increase the fidelity of DNA replication with a factor of about 1,000. And it's a very important factor in preserving genetic information in our genomes. Finally, we can also get damage to our DNA due to external sources like UV light, ultraviolet light, present in ordinary sunlight. And one of the things that can happen is that if we have two T's, the letter T's here in the DNA next to each other, if we get ultraviolet light on these T's, sometimes they can actually be a chemical link formed between them like this that destroys the structure of DNA. This has serious consequences both for the structure and function of the DNA, and we cannot tolerate that.